Good afternoon viewers. Today we are celebrating National Vaccination Day, and it's the best day to define vaccines and to know more about vaccines. And currently with COVID going around, vaccine is one of the current very important discussion happening in all circles. Now, as we all know, today, that is 16th March, is been declared as a National Immunization Day in India. Now, what is immunization? Immunization is a global health and development success story saving millions of lives every year. Vaccines reduce risks of getting a disease by working with your body's natural defenses to build protection. Immunization currently prevents 2 to 3 million deaths every year from diseases like diphtheria, tetanus, pertussis, influenza and measles. Now, as we all know, the three important names who are the pioneers of vaccine development. Dr. Edward Jenner is remembered as the pioneer of smallpox vaccination and father of immunology. Dr. Louis Pasteur, renowned for his discoveries of the principles of vaccination, microbial fermentation and pasteurization. And of course, Dr. Maurice Hilleman, who is remembered as the father of modern vaccinology, as he developed nearly 14 vaccines during his lifetime. Now, how vaccines prevent diseases? As we know, vaccines contain the same antigen or a part of an antigen that cause diseases. For example, measles vaccine contain measles virus, but the antigens in a vaccines are killed or weakened to the point that they themselves don't become an antigen, they themselves do not cause a disease. Now, as we know, there are different types of vaccines. The whole organism vaccines can be where the organism is killed or attenuated, or there can be purified macromolecules from the antigen which is used as a vaccine, like the toxoid or a capsular polysaccharide of the pathogen, or the recombinant microbial antigen or the surface antigen. Now, recombinant vector vaccines, DNA vaccines, synthetic peptide vaccines and multivalent subunit vaccines are the newer forms and the more acceptable forms of the vaccines because of less side effects. Now, the whole organism vaccines, when we are talking about whole organisms live attenuated vaccine, the measles vaccine comes to our mind. Measles is an acute illness caused by a virus of paramyxovirus family. It is transmitted via droplets from nose, mouth or throat of an infected person. The virus infects the respiratory system, making it highly contagious. Globally, an estimated 450 people, mostly children, die every day from measles despite the fact that an effective and a safe vaccine is available and at a very low cost. Now, the first measles vaccine was tested in 1958 by Sam Katz, an infectious disease specialist who was working with Thomas Peebles and other researchers in the Boston lab. He tested the first version of Blab's vaccine on mentally retarded and disabled children at a school outside of Boston. Each of the 11 vaccinated children developed measles antibodies, but 9 also developed a mild rash. That means the vaccine didn't cause full-blown measles, but it did cause the symptoms. The researchers realized the virus used for the vaccine had to be weakened even further. Now, in 1962, attenuated measles vaccine was developed by Maurice Hilleman and his colleagues, who developed it by passaging John Enders measles virus strain over 80 times through different cell types. The resulting vaccine Rubirex was given with a dose of gamma globulin antibodies to reduce reaction, that is the side effects. Now, another important disease, mumps. The mumps is a contagious disease caused by a virus that leads to painful swelling of parotid salivary glands. Mumps mostly occur in children of the age 2 to 12 who have not been vaccinated. Symptoms can cause fever, headache and painful swollen glands in face, neck and the jaw. Hilleman isolated the mumps virus in 1963 from his daughter Geraldine during her illness. Now, it is said that Hilleman's 5-year-old daughter was feeling ill, so she woke him up in the night. He believed that she probably had mumps as she had the typical swollen glands and fever. Though Hilleman was worried for his daughter, yet sensing an opportunity, he drove to his Merck's office to pick up cotton swabs and a vial of broth. Back at home, he swabbed Geraldine's throat and placed the swab in broth and kept it in the freezer. And later, as you can see in this slide, he later used this swabs, the material for his younger daughter, to inoculate it and to protect the younger daughter from mumps. This is how the mumps vaccine was developed. Now, the whole organism vaccine could also be a killed vaccine. In this, I will talk about the anthrax vaccine, the plague vaccine and the polio Salk vaccine. Now, when we are talking about anthrax, one can never forget the 9/11 threat, and these were the envelopes carrying the anthrax spores which were sent to the statesmen or to the congressmen. But because of that, many people in the post office died because of that. It's a biological warfare material. The Bacillus anthracis infection occurs via 3 different routes. Inhalation causing severe flu-like illness with high mortality unless diagnosed and treated immediately with antibiotics such as penicillin, doxycycline or ciprofloxacin can really prove fatal. The cutaneous exposure results in skin lesions with characteristic black deep scars. Cutaneous anthrax has a 20% mortality if untreated, but usually responds to antibiotics. The GI exposure, that is the gastrointestinal exposure, results in ulcers in the ileum or cecum, bloody diarrhea and sepsis, and is nearly always fatal because of difficulty in diagnosis. Now, how the anthrax vaccine was made? It was made by taking a strain of bacterium, that is Bacillus anthracis, and growing it in the laboratory. The bacteria release several harmful proteins called toxins into the surrounding broth. These toxins are responsible for disease in those infected with the anthrax bacillus. The toxins are then inactivated with formaldehyde, so they can no longer cause the disease. The anthrax bacteria are filtered out of the vaccine. Therefore, the anthrax vaccine is made in a similar manner to the acellular pertussis vaccine. There was a plague outbreak in India in 1994. This is the front page of the Newsweek magazine, where plague was rampant in India in 1994. It is caused by Yersinia pestis, a gram-negative bacillus that belongs to the family Enterobacteriaceae. The plague actually bacteria infects the rodents. The rat flea actually bites the infected rodent, and then when it bites the human host, it transfers the bacteria to human. Now, in the bubonic form of plague, the bacteria enters by the skin via flea bite and travels via the lymphatic vessels to a lymph node, causing it to swell. Now, it is also transmitted to humans by inhalation of infected respiratory droplets. The untreated bubonic plague can progress into an infection of the lungs, causing pneumonic plague. Pneumonic plague, however, can be fatal within 18 to 24 hours of disease onset if left untreated. So, early diagnosis and treatment is essential for survival. Now, the first plague vaccine was documented. It was a prophylactic vaccination against bubonic plague, which was developed by Dr. Waldemar Mordecai Haffkine. When the plague pandemic reached Bombay in 1896, Dr. Haffkine became bacteriologist to the government of British India. He soon produced an effective anti-plague vaccine. But recent studies in animals have shown that this vaccine offers poor protection against pneumonic disease. As reported recently in Asian Post in July 2020, there is a suspected case of bubonic plague in the Chinese region of Inner Mongolia. A local herdsman was taken to a hospital after eating marmot meat. The WHO said that it was monitoring the bubonic plague after being alerted by the authorities in Beijing. In the wake of COVID-19 pandemic, many are anxious and concerned at the possibility of the recent bubonic plague reports turning into another health crisis. However, some experts believe that it is very unlikely that this will become an epidemic due to the effective antibiotic treatments that are currently available. Now, coming to the Salk vaccine, Jonas Salk was the one who actually found the polio vaccine. Salk and his team, with the support of National Foundation for Paralysis, began its first test on human of the killed virus polio vaccine. The subjects were resident children in institutions for the disabled and retarded. As described before, Salk tested the vaccine for all the 3 strains of polio, some in combination and some on their own. and he gave this vaccine to his own family also, his wife also, himself also, and his 3 sons also received. This picture actually shows his son receiving the polio vaccine. This polio vaccine results were later announced in 1955 in a press conference, but later the polio vaccination was suspended. Leonard Shield, the US Surgeon General, suspended this vaccination program in order to investigate the safety of all 6 manufacturers' vaccine. The full review concluded that 11 people died from the vaccine and hundreds were paralyzed. Though the cause of the disaster was never proven, it is likely that certain production methods did not follow maybe the SOX instructions and resulted in a failure to completely kill the type 1 Mahoney poliovirus in the vaccine. Now coming to the rubella and the rubella vaccine, Hillman was first one who found out the rubella vaccine, Meruvax, which was licensed. It was later replaced by vaccines derived from Stanley Plotkin's Rh27/3 strain of rubella virus. What does the MMR vaccination actually do? The MMR combination vaccine is against mumps, measles and rubella, and it was first developed in 1971. It was given to the children and it was proved to be highly successful. Now coming to the purified macromolecules as vaccines. Now in this, sometimes the toxoid is being used, like toxin is the metabolite which is produced by a pathogen. This toxin can be inactivated by formaldehyde or something and converted into a toxide. In this case, diphtheria and tetanus are the most important diseases that have been countered by using toxoid as a vaccine. The capsular polysaccharide, for example, in case of Haemophilus influenza type V, its capsular polysaccharide is actually used as a vaccine material. The surface antigen, just in case of hepatitis B, has been used to combat the bacteria, the disease, hepatitis. Toxoids are manufactured from bacterial toxins. Some bacterial pathogens, including those that cause diphtheria and tetanus, produce exotoxins. These exotoxins produce many of the disease symptoms that result from infection. Now, for example, the tetanus vaccine can be made by purifying the bacterial exotoxin and then inactivating it using formaldehyde to form a toxoid. Now, exotoxins are the proteins produced inside the pathogenic bacteria, most commonly gram-positive bacteria, as part of their growth and metabolism. These are secreted or released into the surrounding medium following lysis of the bacteria. Now, another important way of producing vaccines or using a unit or subunit of a pathogen is by using the bacterial polysaccharide capsule. Like for example, in case of Haemophilus influenza. Haemophilus influenza type B, which is called Hib popularly, is the major cause of bacterial meningitis in children less than 5 years of age. Vaccine for Hib consists of type B capsular polysaccharide, which is covalently linked to a protein carrier called tetanus toxoid. The polysaccharide protein conjugate is considerably more immunogenic than the polysaccharide alone. It activates T helper cell, enables the class switching from IgM to IgG, and can cause memory B cell. The proteins from pathogens are also used by recombinant techniques. The gene encoding any immunogenic protein can be cloned and expressed in bacterial, yeast or mammalian cells using recombinant DNA technology. A number of genes encoding surface antigen from viral, bacterial and protozoan pathogens have been successfully cloned into bacterial, yeast, insect or mammalian expression system, and the expressed antigens is used for vaccine development. The first such recombinant antigen vaccine that has been approved for human use is the hepatitis B vaccine. Now, this is hepatitis B. It's a viral infection that attacks the liver and can cause both acute and chronic diseases. The virus is transmitted through contact with the blood or other body fluids of an infected person. More than 7,80,000 people die every year due to the consequences of hepatitis B. This vaccine is 95% effective in preventing infection and its chronic consequences. The Recombivax HP, it's a non-infectious vaccine which consists of surface antigen of hepatitis B virus that is produced in yeast cells. As you can see, the viral RNA is being incorporated into a bacterial plasmid. This recombinant DNA plasmid is then put into a yeast cell, where this recombinant yeast cell multiply and produce more of the antigen in the mammalian host. And then finally, this fermentation, this yeast that is produced in the fermentation tank, the protein which is expressed is used to produce the vaccine. The multivalent subunit vaccine. Incorporation of multiple antigens from the same microbe into the vaccine through construction of SMAA, that is a solid matrix antibody antigen complex, by attaching monoclonal antibodies to particulate solid matrices and then saturating the antibody with the desired antigen. This is an efficient manner or by producing a multivalent subunit vaccine that would lead to a multivalent or a polyclonal sera, which will be much effective in combating the pathogen. Now, many subunit vaccine have been declared. For example, subunit vaccine was formed in 1996 against the tick-borne diseases, in 2011 against malaria, and a peptide based subunit vaccine was also produced against hookworm infection sometime in 2012. What is a DNA vaccine? This is a new type of vaccination. These vaccines contain only portions of the pathogen's genetic material, which actually inserts the human cells to make proteins that elicit an immune response to that particular pathogen. The encoded protein of the DNA vaccine is expressed in the host in its natural form. Now, the DNA vaccines will induce both humoral as well as cell-mediated immunity. These vaccines cause prolonged expression of the antigen, which generates significant immunological memory. Now, a new vaccine that has come up is the cancer vaccine. Here what is done, the tumor cells are removed from the patient and they are placed in the culture. Alternatively, established tumor cell lines are chosen and placed into culture. The tumor cells are inactivated and mixed with dendritic cells from the patient and injected back into the patient as immunogen. But how exactly the cancer vaccine works? In the immune system, the dendritic cells can be used to trigger a response against the cancer cell in various ways. For example, as you can see in the slides, in these two examples, antigens are introduced directly into the dendritic cells or generated in situ using tumor DNA. The cells then produce tumor antigens and direct the T cells to seek out and destroy the cancer cells. Like the emerging infectious diseases outbreaks and their associated vaccine development. The timeline shows how in the last 1 or 2 decades, how new diseases have come up. As you can see, one very prominent disease, the Zika virus, has actually come up in 2015. We are still unable to get a vaccine. Similar goes for COVID-19 that came in 2019. But more importantly, here you can also see an Ebola pandemic that occurred in 2014. That somehow got controlled in 2016, but again reappeared in 2018 and it's still going on, although a successful vaccine came up in 2019. Now, talking of Ebola, as I was telling you, it started in 2014 and again it has resurfaced in 2018. Now, how Ebola occurs? There are 4 stages to it. In the first 7 to 9 days, there are less symptoms, sore throat, fever, headache, etc. In the second stage, vomiting, blood, diarrhea, high fever occurs. In third stage, there is a brain damage, bleeding from nose, anus, mouth. By the fourth stage, the person is in a very bad state and generally death takes place. And this entire thing takes place maybe within 11 to 12 days and the person dies within 12 days or 2 weeks. Now, Ebola virus disease, also called Ebola hemorrhagic fever, is a severe, often fatal illness in human. The virus is transmitted to human from wild animals and it spreads in the human population through human to human transmission through body fluids like sweat, tears, saliva, urine, semen, or even through the bed sheets, clothing, needles, or corpses also, handling corpses. Now, an outbreak in 3 West African countries, Liberia, Sierra Leone, Guinea, from 2014 to 2016 resulted in more than 28,000 cases of EVD and more than 11,000 deaths that are caused by Zaire Ebola virus. Currently, the Democratic Republic of Congo is experiencing the world's second largest EVD outbreak. As of 14th February 2021, that is last month, 4 cases of EVD including 2 deaths have been reported in North Kivu province in the eastern part of DRC, where a large outbreak was declared over in June 2020. 2 health zones are currently affected, Biana and Katwa. In December 2019, the US FDA announced the approval of Ervebo, the first FDA approved vaccine for prevention of EVD caused by Zaire Ebola virus in individuals 18 years of age and older. Now coming to the Zika virus is another virus which is caused by Aedes aegypti mosquitoes which bite during the day. The Zika virus infection has also led to a pandemic. This infection with Zika virus is also associated with other complications of pregnancy and leads to miscarriages. A lot of neurological complications have been found in Zika virus. We are yet to find a perfect vaccine for Zika virus. Now coming to COVID-19. Coronavirus disease 2019, that is COVID-19, is a contagious disease caused by severe acute respiratory syndrome coronavirus 2, that is SARS-CoV-2. The first case was identified in Wuhan, China in December 29. Since then, disease is widespread, leading to an ongoing pandemic. The symptoms of COVID-19 are variable, with almost 81% getting mild to moderate symptoms, while 14% develop severe symptoms, while 5% suffer critical symptoms. Many vaccines have been developed, like for example the Moderna COVID-19 vaccine, the CoronaVac by Sinovac, Covishield by University of Oxford AstraZeneca, Covaxin by Bharat Biotech. Currently, 2 vaccines that have been given emergency approval in India are Covishield as well as Covaxin. Currently, the vaccine R&D map of India can be seen. There are around 26 vaccine R&D institutions in India that are involved in vaccine research. The development of next generation of vaccine will be very challenging, specifically for certain agents like the pathogens like malarial parasites or like the other outwitting pathogen that outwit the human immune response like HIV or influenza virus. In the end, I would quote Nelson Mandela by saying, life or death for a young child too often depends on whether he is born in a country where vaccines are available or not. Hence, we must understand that vaccines save lives. Let us protect our little ones by timely vaccination. Thank you very much.